I'm John Hargrove, and I'm going to be talking to you today about looking at the life cycle of a modeling project. In particular, we're going to be looking at modeling the determinants of growth for an insect vector population. This talk is going to be given in 3 parts. This is part 1. Now, the goals of this presentation, threefold. We want to chart the progress of a research project right the way through from conception through hypothesis, the study design, how the data was collected, the hypothesis being tested, and then up to the paper being published. And then having done that, we're actually looking at a step further where we go from the simple study to add the development of a dynamic model, which is going to use the data from the original study to take things a step further. And in all of this, we want to be stressing the central importance of data and the power of dynamic modeling. Now, first things first, the point is that we're all, as scientists and all human beings really, are trying to make sense of what we see around us. We're always trying to make sense of data. And as scientists, of course, that's what we're trying to do. And there are lots of different ways of doing this. We are always liable to be looking for patterns in the numbers that we've been collecting. And we can do this in a lot of different ways. For instance, by plotting graphs, by making tables, by doing statistical analysis. And those fairly simple, straightforward things can be perfectly sufficient to decide if the hypothesis that we're trying to test is correct or whether it's not correct, and may be perfectly adequate to allow us to publish the data. But sometimes this simple process doesn't give the whole story, and it may fail to capture some of the dynamics in the system that we're actually looking at. Now, a student on the course in past years actually asked some useful questions regarding this whole process, and asked, for instance, what kinds of data are useful for modeling? How to collect or access the data that we're going to be using, network data for contact patterns, weather data, disease incidence data, that kind of thing. And also asked, is it okay to take data or parameter estimates from other studies and use them in your own model? We will think about that as well. And finally, if data is supposed to be for epi-orientated people who are here to learn about mathematical modeling in order to collaborate with modelers and speak their language, it would be helpful to know how we can actually use our epi methods training to contribute to the model development. Well, I hope by the end of this talk, we'll be able to answer those questions, but we'll just bear them in mind as we go along. The question is, why would we want to model at all? And one reason is that we've got a problem that we can't actually solve any other way. We've tried doing the graphing and the tables and the statistical analysis, but there is something that we don't understand. We don't know how to get at it. Also, we might learn something from the modeling that we couldn't otherwise learn. And also to learn actually what we're missing. If we have gone through this study and we actually do the modeling, we can see, okay, this is what further work we need to be doing. And also, sometimes there are more interesting things in the data than we actually, we developed a particular project in a particular way to answer particular questions. But actually, when we actually did the work, then suddenly all sorts of data came out of it, which were actually really interesting and opened the doors for doing other work. Now, the particular work that I'm going to be talking about concerns the tsetse flies, on which I've worked for the last 50+ years, particularly in Zimbabwe. We're going to be looking at one species of them in Zimbabwe. Tsetse flies are still pretty important. They occur over 11 million square kilometers of Africa, and they transmit trypanosomiasis, which is sleeping sickness in humans, very much in West Africa and Central Africa. And also they transmit nagana in livestock, which is an important problem in East and Southern Africa. Very unusual flies. The female doesn't lay lots of eggs like a mosquito. She just produces a single larva. But you can see from this that this larva is a beast. it can weigh as much or as more than the mother that's just produced it. So she only produces one every sort of 8 to 10 or 12 days, depending on the temperature. So a very low reproductive rate. So when the life cycle is basically that the larva develops inside the mother, as we saw, it is then produced. It burrows under the ground and forms a hard pupaриal case and just sits there for 20 to 40 days, doesn't feed at all, just uses all the energy and raw materials it got from its, and then pops out 20 to 40 days later as an adult, and then the life cycle is complete when the female ovulates again and we have another larva. So there are the pupae that were sitting under the ground here, and what happens is that metamorphosis actually occurs inside this pupaриal case, and ultimately what pops out is a fully developed fly, and a few minutes afterward that it's emerged, it looks like this. Okay. So this experiment was done in Zimbabwe. Here's Zimbabwe, you know where it is. And if I blow it up, there's Harare, and here's Lake Kariba on the Zambezi River. And we're going to be looking at an experiment done on an island just off the town of Kariba. Now, the experiment was actually designed by Glenn Vale, with whom I've worked for the last 50 years. And the important thing that Glenn did was to find that tsetse flies use odors to find their host. That's how they actually get to feed. And what he and I and others have done is to actually isolate those odors. And then the question arose, well, if we can do that, perhaps if we put them out at sufficient concentration, we could actually pull in sufficient numbers of flies to actually use it to control or even eradicate tsetse flies. And so that was the hypothesis, that we could possibly do this. So what he wanted to do was to have a situation where you could actually test this. And an island is a very, very good way of doing it, basically because islands are closed to in-migration and out-migration. You've got a chance of actually knowing how many flies you've got on the island and knowing whether, in fact, you've actually got rid of them all. These are the kinds of devices that he was thinking of using. Here's a trap. Here the flies would come in here and get caught in the cage up at the top here and would die. But a much simpler thing is actually just to have a single sheet of cloth, blue, sort of they are attracted to the blue, but they actually land on the black. So if you put some insecticide like deltamethrin on this cloth, then the flies land on it and they die. And the question is, if we were to use, quite small numbers of these targets or traps on an island, could we actually use it to control or get rid of, eradicate the flies on that island? So here is Antelope Island, which lies just off Kariba town there. Antelope Island, it's about 5 square kilometers. When we went there in the late 70s, there were no flies on there, and there were very, very few wild animals, just a few impala and baboons. There weren't any flies in it. So basically what Glenn was to put on 4,000 pupae, and also introduce 33 cattle, put them in fenced paddocks, so that there was lots of things for the tsetse to feed on, because all they feed on is blood. Both the male and the female feed only on blood. Now you've got an island and you've got tsetse flies and you've got food for the tsetse flies, but now what you need to do is to allow this population to grow, and you need to know how many there are, so that you know how many, and then you can see whether you can actually take it from the number that you've got down to 0. And the way that you actually estimate how many flies you've got is using a method called capture, mark, release, recapture. So what happens here is you send an ox out, a couple of men with the ox, and they catch flies using a hand net. And then once you've caught it, and you take it out of the net here, and then you mark it with a little dot of artist's oil paint on the thoracic surface, and the position that you mark it on and the color that you use denotes the week in which the fly is captured. And then you do that again and again, and you record the flies which have been caught before, and you can use that to actually get estimates of the population. And this was a a very extensive program. These fly runs, as they were called, were done 4 times a day, every day for more than 4 years. And it's probably the most intensive mark release recapture study ever carried out on any insect. As a consequence, we got very, very good population parameter estimates. In particular, we could tell how many flies there were on the island on each week of the experiment. So this is across the whole 4-year period. Every week, we could get an estimate of how many flies there were on the island. Blue for males and red for females. And you can see that they're really quite good estimates. Now, what happened was that Glynn allowed the population to grow for a couple of years until the end of 1981, and then started putting on traps, trapped in the morning for a while, and then trapped in the morning and the afternoon later on. And then ultimately what he did was to put the odor-baited targets on there, only 4 per square kilometer, and the population just crashed and crashed and crashed, and ultimately it actually went extinct in around about Christmas 1983. So we could actually look also at the survival probability of the flies and also the numbers that were born. Here's the survival probability. As you can see, the survival probability is lower for males than females, a bit like humans. And and what you also see, which is interesting and which we'll come back to later, is that you see an annual cycle that the survival probability goes up and down. Of course, later on, when you've got targets on the island, the survival probability is very, very low. But even when you didn't have any targets on there, there was a cycling. And what happens is when it gets very hot, this is the temperature down here, it gets very hot in October, November, the survival probability is low. When it gets cooler in June, July, then the survival probability is rather higher. So we got all that information. Now, the point was that, as I've said, what Glynn did was to allow this population to grow to tremendous heights. At one point, there were 30,000 flies on the island, and he was able then actually to eradicate the flies just using 4 targets per square kilometer. And this gave rise to a tremendous industry, basically, because people realized we can use this as a much cleaner way of getting rid of tsetse flies. In the past, we've had to shoot wild animals or spray DDT or deodorant across huge amounts of area. With this method, all you've got to do is have 4 of these devices per square kilometer, small amount of insecticide, which is only on the target, not on the bush or anything like that, and you can actually do the job. So that was really great. So he's answered the question that he set out to answer. Can we actually use these targets to get rid of tsetse flies? The answer is yes. But now, here's the interesting thing. We've got, as I said in the lead up to this, we've got a situation where we know what's happening with the numbers. We know what's happening with the survival probability. But the question is, what's driving this? Is it purely the changes in temperature, other than the use of targets, which are causing the population to go up and down? What is it that's actually affecting population growth? Is it simply the temperature, or is it the humidity, or is it other things? And we can't answer that on the basis of just drawing the kind of graphs that I've shown you. So what we need to do is now we've got to the point where, yes, we can see we need a model. We want to model the growth and the decline of this Glossina morsitans population on Antelope Island. So now I want you to take a break, and you can start looking at part 2 as and when you feel like it. But while you're taking your break, you might like to look at the paper that was actually published on this work, and then I'll see you later.